Hello, everyone. Let's go ahead and get started. My name is Karen Dillon. I'm the Executive Director at the Cancer Vaccine Institute, where we are developing approaches to ignite the immune system to end cancer. We are grateful to all of our patients, collaborators, and donors for their support of our research and mission. It's my pleasure to welcome you to the second session of the UW Medicine Cancer Vaccine Institute's Summer Seminar Series. Just a little housekeeping, if you could keep your phones on mute. The duration of the talk, there will be opportunity to ask questions at the end, hopefully if time allows. Today's presentation is on using the immune system to treat cancer by Dr. Nora Disis. Dr. Disis is the director of the Cancer Vaccine Institute. She's also the Associate Dean for Translational Health Sciences at the University of Washington, Professor of Medicine and Adjunct Professor of Pathology, Obstetrics, and Gynecology, and the Helen B. Slonaker Endowed Professor for Cancer Research. She's also the editor-in-chief of JAMA Oncology and an expert in tumor immunology and translational research. She's one of the pioneering investigators who discovered that HER2/neu is a tumor antigen. So without further ado, I give you Dr. Disis.
Thank you very much, Karen. I'm going to start where Andrew left off last time, and I'm going to be talking about the adaptive immune system. Now, unfortunately, I'm having a problem moving things forward. Let's see if I can figure this out. Here we go. I'm going to talk a little bit about the adaptive immune system, really composed of T cells and B cells. I think one of the biggest breakthroughs in understanding how to use the immune system to treat cancer actually came with us understanding what is clinically effective anti-tumor immunity. And this didn't come from treating patients with different drugs. It actually came from looking at lots and lots of tumors using microarrays and counting the immune cells in the tumors and trying to figure out which immune cells correlated with clinical outcome. And this is just an example taken from a review article that if you had a very high density of type 1 T cells, and Andrew talked about this last week, CD8 cytotoxic T cells or CD4 T helper cells that secrete type 1 cytokines, and their memory cells, you are more likely, if you have a small tumor, not to relapse after 15 years. And if you have a very large tumor, you're going to be someone who has a prolonged survival compared to others at your same stage. And in fact, for many tumor types, melanoma, colon cancer, breast cancer, we've been able to make an immune score where we can actually quantitate the number of T cells that you need to have a better prognosis. And these immune scores are now working themselves into the standard staging system of many types of cancer. So again, a high density of cytotoxic and type 1 T cells of a memory phenotype, meaning your own body stimulated them, that are able to penetrate deep into the tumor. So T cells need to respond to an immunogenic protein. And Andrew talked about this before. What does the immune system see in cancer? And it used to be, we didn't think cancer was very immunogenic, but now we know that there are many proteins that stimulate an immune response in cancer. The majority of those proteins are very weakly immunogenic. They're normal proteins that are related to the growth of cells that have become markedly upregulated when cancer turns on. So your body knows that there's something different about these proteins and tries to make a response to them, but not the type of response that we know is needed to kill cancer, those cytotoxic T cells. We do know in some patients, the immunogenic proteins in their cancer do give a foreign signal. And that occurs in patients whose tumors are caused by a virus, such as cervical cancer and HPV, or hepatocellular carcinoma and hepatitis B, or they have lots and lots of mutations. And those mutations have never been seen by the immune system. What do we know about the immune response when you're responding to aberrantly expressed self or these foreign signals? Well, the immune response that's generated to these self antigens, which really are the most common antigens in cancer, is called a type 2 immune response. It largely consists of antibodies and T cells that secrete cytokines that prevent the generation of type 1 T cells. And these may be called cold tumors because they don't have a lot of T cells in them. This are things like breast cancer, most colon cancers, pancreatic cancers. Those that are highly mutated have lots of type 1 T cells. They're getting that foreign signal, that danger signal. These are tumors like melanoma, lung cancer, and virally induced cancers and genetically unstable tumors, which I'll talk about in a second. So, how many cancers actually have a lot of virus, a lot of mutations? And we can calculate the number of mutations you need in your cancer to be able to have it recognized by the immune system and create that type of type 1 immune response. And you can see on this far left-hand panel, melanoma, bladder cancer, lung cancer. These can be highly mutated cancers, and the majority of patients do have the number of mutations needed to potentially stimulate the immune system. Whereas if you look at breast cancer, um, brain cancer, ovarian cancer, you can see the majority of these patients have low to no mutations. And this is looking at over 7,000 tumors, almost 8,000 tumors from the TCGA project. So you can see that those patients who have the potential to have this type 1 endogenous immune response is really restricted to a few cancer types. And in fact, this is data that was taken from the Cancer Immunity Project, again, using the TCGA. If you look at the two cold tumors, breast cancer, and the majority of gastrointestinal cancers, you can see that when you evaluate their T cell infiltrate in their tumors using genetic markers, the majority of that T cell infiltrate is that Th2 infiltrate, and only a minority of that infiltrate is that Th1 infiltrate, that type 1 infiltrate. And in fact, looking at Th2 versus Th1 turned out to be a very important prognostic factor across all cancer subtypes. So, how do we use this data and develop immune therapy? Well, one of the first types of immune therapy that showed any efficacy in a large number of patients are what's called immune checkpoint inhibitors. And I'm going to talk about these now in terms of some of the clinical trials. This is not going to be an exhaustive talk, but what we have learned about using immune checkpoint inhibitors. So what is an immune checkpoint inhibitor? Well, you can imagine you have a T cell in your body that's capable of killing things. There has to be a lot of checks and balances by your immune system to turn on your immune response and turn off your immune response. So we have a lot of activating as receptors on our T cells that, and these are shown in blue here on the left-hand side of the slide, that give that signal that say, turn on, attack, proliferate. There's something here you need to respond to. And we also have these checkpoint inhibitors, and this is shown in the red, and there's several of them. CTLA4, which I'll talk about, PD1, which I'll talk about, but many more, which are also becoming antibody therapy that's being tested in cancer. And these checkpoints tell the immune system to turn off. for many reasons. The cancer becomes inflammatory. These immune checkpoints become upregulated so that your body can turn off that inflammation. We don't want that. We want inflammation to go on. The antigens or immunogenic proteins are recognized as self. So these checkpoint inhibitors, these immune checkpoints become upregulated because your body is getting the signal, no, no, don't respond to this. This is part of our self. So the very first successful treatments using solely the immune system to treat cancer blocked these checkpoint inhibitors. And the antibodies blocked CTLA4, and I'll talk about this in a second, and PD1 or PDL1. So you have these receptors that are upregulated on the surface of T cells or on the tumor that are telling the T cell stop responding. So researchers made antibodies that blocked those receptors so the T cells did not get the signal to stop responding. And we have several anti-PD1 monoclonal antibodies, nivolumab, pembrolizumab, most common, and we have some PDL1 monoclonal antibodies. This is the receptor that's on the surface of the tumor or innate immune cells, avelumab, atezolizumab, and then the anti-CTLA4 monoclonal antibodies such as ipilimumab. Now, for these to work, you have to have T cells in your tumor that are capable of expanding and killing the tumor. If you don't have those T cells in the tumor, there's nothing really to unleash. And often people talk about these immune checkpoint inhibitors as unleashing the immune responses that is there. So you have to have an immune response there to unleash. So let's go into a little bit of detail about the two groups of immune checkpoint inhibitors, those that target CTLA4 and those that target PD1 and PDL1. What's the difference between the two? Now, this is a little complicated of a picture, but it really tells the story of the difference between CTLA4 and PD1 and PDL1. CTLA4 was the first immune checkpoint inhibitor developed, and it works by allowing more efficient priming of the T cells in the lymph node. What do you think about your lymph nodes? Let's say you have a terrible sore throat like I have today, and your lymph nodes are kind of enlarged. That's because you have a lot of these innate immune cells, such as monocytes or macrophage or dendritic cells, that are trafficking, picking up the infection and going to the lymph node where the T cells are hanging out and saying, alarm, you have to get out there and start killing this infection. So this is called priming of the T cells, the initial interaction between T cells and the innate immune cells. And so what these immune checkpoint inhibitors do is they regulate the amplitude of this early activation, not only of memory T cells that are hanging out in the lymph nodes, but also naive T cells. So they haven't yet seen their antigen. So it's really a huge signal in the lymph node to unleash lots of different T cells. They may be specific for the cancer, or they may not be specific for the cancer. So we have several antibodies, like I told you, targeting CTLA4. Now, out in the tissue, in the cancer, there is another immune checkpoint system. This is called PDL1 and PD1. PD1 is expressed on the surface of a memory antigen experienced T cell. It's already seen its antigen. It's ready to attack. And when there's a lot of inflammation, or the antigen gives that self signal, PDL1 or PDL2 becomes upregulated and tells the T cell, don't attack, don't do this. You've got to stay in kind of a sleeping or anergic state. So this prevents T cell expansion in the peripheral tissues. And it's more, PD-1 and PD-L1 are more broadly expressed than CTLA-4. It's kind of our line of defense against peripheral autoimmunity. So you can imagine antibodies against either PD-1 or PD-L1 will prevent that blocking and allow those T cells to expand. And in general, when you think about where the mechanism of action is, CTLA4, which is a central mechanism of action, is generally associated with a lot more toxicity because it's expanding a lot of T cells, tumor specific, tumor non-specific. Whereas the PD-1, PD-L1 monoclonal antibodies have a little less toxicity because they're really allowing the expansion of tumor-specific T cells. So we have heard about immune checkpoint inhibitors. We've read about them in the Wall Street Journal. They've been touted as the cure for cancer. What really is happening with immune checkpoint inhibitors? Well, here you can see on this left-hand side of the slide, in some tumors, they really do seem to be a magic bullet. When you see Merkel cell carcinoma or melanoma or bladder cancer, we're getting response rates of 70%, 50%, 40%. And we're really seeing complete response rates, meaning the tumors go completely away in about 10% of patients. So they're super effective at controlling cancer growth, and they can be effective over a long period of time. But here you see in some of those cold tumors, gastric cancer, cervical cancer, we're really seeing response rate about 10%. If you look at things like most breast cancers, most ovarian cancers, again, we're talking about response rates of about 10% with very few complete responses. So despite what you read in the newspaper, it isn't the complete magic bullet, but the immune checkpoint inhibitors are definitely working their way into the armamentarium of what we use to treat cancer. And this is just an example of all the FDA approvals for immune checkpoint inhibitors. Triple negative breast cancer, they are used in gastric cancer, advanced renal cell carcinoma, hepatocellular carcinoma, and many of these are used in combination with chemotherapy. Chemotherapy to control tumor growth, the immune checkpoint inhibitor to unleash whatever T cells are capable of killing the tumor. So we're still working on trying to get immune checkpoint inhibitors to work for most cancers. And there are many new drugs that are coming down the pipeline. And I'll share a little bit about the thinking of how to make these work better for most cancers. But before we move on from immune checkpoint inhibitors, I want to show you a little bit about what we're learning to choose the people most likely to respond to immune checkpoint inhibitors. Now, why should we do that? Why should we not give immune checkpoint inhibitors to everyone, even these people who get like a 5% response rate? Well, one of the reasons why we don't is that they are toxic drugs. Um, I think in general, from my standpoint, for the majority of patients, they're less toxic than chemotherapy, but for a minority of patients, they can be quite toxic. And basically, what is the toxicity? They're called immune-related adverse events. People can develop colitis. They can develop very significant autoimmune hepatitis. Many people, about 20%, develop skin rashes or pruritus. Endocrinopathies like thyroiditis or very significant problems with the pituitary or inflammation in the lungs. Now, The vast majority of these toxicities are mild and they can be managed with drugs or they can be managed by stopping the immune checkpoint inhibitor, letting the patient recover, and starting the immune checkpoint inhibitor again. And you won't necessarily get the toxicities back again. But in a minority of patients, less than 20%, the toxicities can be very, very significant. And the toxicities differ by drugs. So you can see anti-CTLA-4 causes a different pattern of toxicity, ipilimumab, than something like nivolumab or pembrolizumab do. And the type of toxicities come up at different time frames, and they can be quite delayed. And we even use both these checkpoints together in some patients. And when you're on a regimen that is using a combination of the CTLA-4 directed monoclonal antibody and P1 or PD-L1 directed monoclonal antibody, the toxicities are more and they're more significant. But again, we only use the combination when it shows the patients have a significant survival benefit. So it's because of these immune related toxicities that we're really trying to use biomarkers to hone in on what patients really will benefit from immune checkpoint inhibitors. Now, obviously, one of the first biomarkers people looked at was the expression of PD-L1 on the surface of the tumor or on the innate immune cells in the tumor. And generally, we measure PD-L1 expression by staining. So here you can see a normal cervix. Here you can see a cervical cancer that highly expresses PD-L1, and you can see this brown staining is the antibody that marks PD-L1. And we create what's called a combined positive score. We found that you can have high expression of PD-L1 on your tumor, and you can have high expression of PD-L1 on the immune cells in the tumor. And when you add those together, you really get a good sense of PD-L1 expression. And here you can see a publication of almost 10,000 patients. These are all different types of cancer that are showing you the level of PD-L1 expression, the percent of patients that have at least 1+ expression. That's the level we choose to call positive PD-L1. And again, you see there are winners in this and there are losers in this. But if you look at diseases like cervical cancer or bladder cancer or anal cancer, immune checkpoint inhibitors have become part of the standard of care of treating these patients, mostly due to their one expression. Whereas if you look at things like colorectal cancer, cholangiocarcinoma, prostate cancer, we still are not using immune checkpoint inhibitors in the treatment of these diseases. But there are other markers that we can look at in tumors that will predict response to immune checkpoint inhibitors. And one of these markers is called mismatch repair deficiency or microsatellite instability high. Now, what does that mean? Well, you know, in your body, cells are dying all the time, and we're constantly replicating cells. And your body has tons of mechanisms to correct any errors that occur during that replication. But sometimes in tumors, your machinery that corrects these errors falls apart. And the machinery does not repair these alterations that are occurring when you're replicating your DNA. And so what happens is that you get a lot of unstable DNA that is propagating these errors, and many of these errors end up encoding mutations or abnormalities that can be recognized by the immune system. And I'll talk a bit about that on the next slide. So if you look at this on the right-hand side of the panel, this is almost 12,000 patients, they're tumors. And you can see again, we have tumors like brain cancer or breast cancer or ovarian cancer or pancreatic cancer, where we really don't see much DMMR or mismatch repair deficiency. But we have cancers like endometrial, which was never thought to respond to immune checkpoint inhibitors. In fact, they did clinical trials with endometrial and it fell in that kind of 10% range. Gastric carcinomas, where a significant number of patients in their tumors show this mismatch repair deficiency that may lead to multiple mutations. So let me give you an example of a clinical trial, again, in ovarian cancer, where they enrolled patients that they had pre-screened and saw that their tumors were positive for mismatch repair deficiency. So they had deficiencies in the genes that repaired DNA replication. So they enrolled 71 patients that were chosen by the biomarker. And what they saw was incredible. Instead of an overall 10% response rate, they had an overall response rate of 30%, with almost 13% of patients having complete responses. And this study was called the Garnet trial. And here you can see the patients' complete responses. And even in the 30% of patients who had partial responses, or the 15% of patients who had stable disease, the stability of their disease went on for a very long time. Here you can see almost 69 months. People may have cancer, but the cancer is not growing. And this is just by finding a biomarker, DMMR, that predicts those patients most likely of responding to immune checkpoint inhibitor, because they probably have T cells in their tumor already that have that killing phenotype. Another marker we know is called tumor mutational burden. And if you remembered back to one of the very first slides I showed you of all the tumors and which ones are most likely to have mutations, that number that I cited to you, that is the level you need to probably think that you have T cells that are responding to this, the tumor, is called the tumor mutational burden. And so to get into a little bit more detail about what's happening, the tumor cells with high mutational burden have mutations that have never been seen by the immune system before. The problem is you need a lot of mutations to get increased T cell reactivity, because the mutations are seen as foreign antigens, and that type of interaction to create cytotoxic T cells is a very restricted interaction. So if you have one or two mutations, the chances of you being able to process that mutation in your immune machinery to create an immune response is very, very low. So you need lots and lots of mutations. And that's not necessarily a good thing. Before immune therapy, people who had lots of mutations in their tumor, it generally causes the tumor to grow faster. But now we know having a lot of mutations in the tumor also is immune stimulatory. And so this MSI high, or microsatellite instability, and tumor mutational burden kind of go hand in hand in terms of being able to predict patients that respond to the immune checkpoint inhibitors. And again, this translated to the clinic. This is a very early study. It's called Keynote 158, where it enrolled all comers with all different types of cancers. And here you can see on the left-hand side of the slide, these are cancers that never really responded very well to immune checkpoint inhibitor therapy. Like I said, endometrial cancer was like a 10% response rate, etc. But when the patients were enrolled, they did, in this Keynote 158 study, lots of biomarkers. And then when the study was done, they went back to the biomarkers that they prospectively collected, and they asked the question, did having this marker in the cancer predict whether you would respond? And here is a tumor mutational burden high, tumor mutational burden low. It was a huge study. You can see 619 patients compared to 91 patients. But look at the response rate here. This is the change in their cancer for the negative. And here you can see several of the patients, their cancer totally went away. Whereas if you look at the non-tumor mutational burden high patients, many less of them had a response. So here you can see complete response rate, 4%. It was all in the TMB high. Partial response, most of it was in the TMB high. So we now have these markers. If you have any type of cancer that's dMMR positive or TMB high, no matter what your cancer is, you can get an immune checkpoint inhibitor. And this is the first approval for drugs based on a biomarker. They're tissue agnostic. So if I have a breast cancer, hormone receptor positive breast cancer, immune checkpoints are not approved for that. But my doctor looks at my tumor mutational burden and it's high, I would be able to get an immune checkpoint inhibitor. So it's very, very exciting that we're really using molecular medicine to hone in on patients most responsive to immune therapy. But while not everyone responds to immune checkpoint inhibitors, there are many other immune therapies that are coming down the pipeline or are being tested or have been approved. And I want to talk about a group of immune therapies called T-cell therapy. Now, why do people focus on T-cell therapy? It's because we know one of the problems in many cancer patients, as I've said earlier on in the talk, is they simply don't have the right T cells in their tumor to be able to even unleash with an immune checkpoint inhibitor. So can we give the patients T cells? And we do this. There are approved therapies using T cells. One of the approved therapies is donor lymphocyte infusion. If I get a bone marrow transplant from my sister, and I end up recurring my disease, I can take my sister's T cells because basically my immune system has been replaced by my sister's immune system. So the only cells in there that would be my cells would be the cancer cells. And I can give the donor lymphocyte infusion from my sister, an infusion of my sister's lymphocytes, and they will attack the leukemia that's growing back. And basically, we do see some responses, and this is part of the standard treatment. They're not 100%, and I'll show you how we've moved on. There are clinical studies looking at lymphokine active killer cells. I'm not going to get into that too much. People have taken lymphocytes directly from the tumor, so we know that they're probably responding to antigens in the tumor. They're growing them up in great numbers outside the body, and we're infusing them back into the patients, we do see some responses. The responses in general are lower than what we see with immune checkpoint inhibitors. But where we've really seen gains in adaptive T cell therapy is through engineering the T cells. So actually taking the patient's cytotoxic T cells and taking them outside the body and manipulating them to specifically recognize the tumor. And I'm going to show you an example right now of chimeric antigen receptor therapy. And this is an approved therapy for several types of cancers. And so basically what investigators have done is they've taken a killer T cell, so we already know it kills, and they've put a linker on the outside of the T cell that it's an antibody specific for the cancer. And many hematopoietic malignancies um, uniquely express on every single one of the cancer cells a target protein like CD19. These are expressed on B cells. It's part of a B cell. You have to have CD19 on your B cells. That's what defines a B cell. But B cells can become leukemia. They can become non-Hodgkin's lymphoma. They can become acute lymphoblastic leukemia. So you're basically taking a killer T cell from the patient, you're making it recognize the B cell, and you're adding one of those activating co-stimulatory receptors that I talked about on one of my earlier slides. I had said we have checkpoints that turn off the immune response, and we have activators that activate the immune response. And the second generation chimeric antigen receptor T cells only used one activator, but the third generation CAR used two activators, and this made them extremely effective. And they can see response rates in 90% of the patients. And these have been very effective treatments. Taking these T cells, um, manipulating them outside the body, and then infusing them back into the T cell, into the patient. So these are highly activated, highly active T cells that bind with great affinity to CD19 for the most part, but also we're seeing approvals in multiple myeloma to myeloma antigens that are expressed on the surface of those tumors. But like every immune therapy, CAR T cells do have their challenges. They haven't shown much efficacy for solid tumors where there are many antigens and where you don't have this clonal population that all have the same antigen. Sometimes you can eradicate the cell, but other cells will grow through. So this is kind of the generation of antigen negative variants. Well, we also know that the T cells have been engineered, so they're not totally natural, and they don't last that long in the peripheral blood. So one of the problems with CAR T cells is that they go in, they do their activity, and they die. Whereas if you can have your own endogenous immune response, that would last much longer. They also have only shown efficacy in blood cancers because they have a hard time penetrating solid tumors, which are a ball. And so they may get to the solid tumor, but they don't necessarily break in, which, if you remember on the first slide I showed you, is one of the hallmarks of a clinically effective immune response. And finally, for solid tumors, the tumor itself is secreting a lot of substances that dampen the immune response. And you don't see that with liquid tumors like leukemia. So lots of potential for leukemias and lymphomas. We're still struggling to make them work better for solid tumors. And again, The CAR T cells are not without toxicity, some very significant off-tumor toxicity. They can totally deplete all your B cells, even the ones that aren't cancer, and that can leave you prone to infection. There's cytokine release syndrome. Your immune system gets turned on so much, you become very sick. You can have an allergy to some of the proteins used for the engineering, and you can also have some neurologic toxicity that people are still figuring out why this happens. But for those patients where these drugs are approved, they can be life-saving. And again, more and more people are doing the same thing for CAR T cell therapy as we did for immune checkpoint inhibitor therapy, trying to figure out which patients will benefit the most. So I think right now, most of the strategies that people are taking into combination with either immune checkpoint inhibitors or CAR T cell therapy are really focused on increasing T cells. And I have a tendency to break patients up into two types. They have T cells present, in which case you'd want to start with an immune checkpoint inhibitor. Maybe you'd like to try to activate those T cells more. Maybe you would try to condition that microenvironment by blocking some of these substances the tumors are secreting. There are more than one way to unleash the T cells. But we do know for most patients, T cells are needed. And adaptive T cell therapy is one way to give them back. I'm going to talk about two other ways to end the talk: vaccines and turning the tumor into a vaccine. So when we think about vaccines, I mean, we've all lived through COVID. You go get your COVID shot. What you don't, many people don't understand is that vaccines are very complicated, multifaceted drugs. They consist of the immunogenic protein that you're trying to immunize against, a delivery system for that antigen or immunogenic protein, and then all vaccines usually have an adjuvant or a non-specific immune stimulator to give that vaccine a jump start. When I started looking at cancer vaccines, one of the biggest questions were, what were the antigens? What were the immunogenic proteins? And we have learned a lot. Proteins become upregulated and immunogenic all through cancer. And much like CAR T cells, you have to choose those proteins that, if you can eradicate cells that have upregulated these proteins, you're really going to have an effect on the cancer. So we're really making molecularly targeted vaccines specific for certain types of cancers. And there are many delivery systems, loading them onto antigens onto our own autologous antigen presenting cells, delivering the antigens in viruses to give the danger signal, pre-digesting the antigens specifically for the immune system. This is peptides, DNA or mRNA, as we've seen with the COVID vaccine, also gives a very potent danger signal and stimulates the right type of immune response, that type 1 immune response. And there are many adjuvants that we use to further give that danger signal. And this is just a list of them. They're very well studied, and they work with the delivery system to try to create the highest type 1 immune response possible. Now, many people say vaccines have not really been effective. And this is a recent meta-analysis of many types of vaccines for breast cancer. And there's very similar data for ovarian cancer. This paper was about those two cancers that are cold tumors, and there's not an approved vaccine. And these investigators asked the question, is there any activity whatsoever? And these studies went back for many, many years. They all used vaccines as a single agent in metastatic patients. These are very advanced stage patients who were heavily pretreated with chemotherapy. So you would think they probably don't even have a chance to respond to a vaccine. But what these investigators showed was very interesting. Most clinical trials really showed some type of response. Again, these were clinical trials chosen, 428 patients, where they were asking the vaccine to shrink the tumor a little bit. And overall, they had a 10% response rate with just the vaccine alone. Now, if you go back to the data I showed you about immune checkpoint inhibitors for breast cancer, in general, they were about a 10% response rate. So it was a very interesting study because it showed us there is activity using a vaccine in breast cancer. And when they looked at those studies, there were 10 trials that really had long-term survival in this heavily pretreated patient. You're talking about 180 months. You still have about 30 of those patients still alive. So this is using old technology in vaccines. So I think the good news is there's nowhere to go but up in terms of vaccines because we're in a new era of cancer vaccines. I told you there are two types of antigens. One are these mutation antigens, and many investigators are focusing on trying to look at the tumor mutational burden, finding mutations specific to the individual patient, and tailor making a neoantigen mutation specific vaccine. And there are many clinical trials of this approach. But other investigators in our group, I think, are the leaders in this, have really focused on those self antigens, which are the most common antigens. And what we found was within the natural protein sequence of these self antigens, we could identify epitopes that interacted with human class 2 molecules that stimulated type 2 immunity and some rare epitopes that selectively only stimulated type 1 immunity. And so we're now editing self-proteins to create Th1 selective vaccines that do not stimulate any evidence of a type 2 response. And these are in clinical. I just want to show you an example. This has been published quite a while ago of a HER2 ICD vaccine in metastatic breast cancer. And it shows you a few things. Number one, HER2 is a very important protein in about 30% of breast cancers. It's a driver oncogene. It's a reason why patients have their disease. And we looked at a phase 2 study of about 22 patients that had metastatic disease, so they had stage 4 advanced stage disease. 70% of these had been treated with greater than 2 previous lines of chemotherapy. And as you can see, much like people wondered in that meta-analysis I showed you, the patients could respond to a vaccine. They went from very low T cell precursor frequencies to very high T cell precursor frequencies to the HER2 epitopes. And you can see 60–80% of patients were responding to multiple class 2 epitopes derived from HER2, as well as the entire intact extracellular domain and intracellular domain protein. When you looked at these stage 4 patients, their progression-free survival was about 40% at 50 months. So many of the patients were not developing their disease again, and their overall survival was 80% still alive at the time the study ended, 50 months. Now, the question is, do these T cells home to the tumor? And one of the things we did in one of the patients who did get their disease back again, we took their T cells, and since they had a high level of T cells in their blood, we were able to easily expand them ex vivo and then infuse those T cells back into the patient as an adoptive T cell therapy. And here you can see this patient who had bony metastases pretty much throughout her body. When we labeled these T cells with indium, you can see that they homed to the site of metastases in the shoulder, in the cranium, in the sternum. And when we did rapid PET imaging, looking at glucose uptake in the site of her metastases over a 48-hour period of time, you can see that the T cells were getting into her tumor. This is the percent increase of SUV. So all of a sudden, these tumor sites went from being cold to hot. And this is one of the things we see with vaccination. Many other groups have shown that vaccinated T cells are much more activated than endogenous T cells that are educated on the tumor. The final two slides I really want to talk about are using the tumor itself as a vaccine. And one of the drugs that's been approved, it's not super widely used, is oncolytic virus therapy. It's mostly used in melanoma. And what that is you actually infect the tumor with a virus. The virus gives a potent danger signal. And as the infection is spread, the tumor cells are killed. Lots of inflammation is caused, that kind of type 1 cytokine type inflammation, because this is a virus. The tumor antigens are released, and then you get systemic anti-tumor immunity. And this is a study that was published a few years ago that looked at patients with melanoma that had already had immune checkpoint inhibitor therapy, and they either had not responded or recurred their disease. And you can see here that they were able to get 38% complete response by using the oncolytic virus therapy and giving back pembrolizumab, an anti-PD-1. So the response rate was 48 to 89 was 71% in these patients with previously immunotherapy modulated melanoma. And people are really working to see how to make oncolytic virus therapy work better for other diseases that you can inject, let's say liver metastases or hepatocellular carcinoma. This last example is something really simple, and that's looking at T cell priming cold tumors, and this is a case of breast cancer. About 20% of breast cancer patients will recur their disease in their chest wall, and this is an example of a patient who's got chest wall disease, and you can see a biopsy of that shows lots of different tumor nests. Lube Salazar in our group used a topical toll-like receptor ligand called imiquimod and treated these patients with chemotherapy, which really is associated with less than a 15% response rate for chest wall disease, because many of these patients have already had radiation therapy and their blood flow is really impacted to the tumor. So she gave topical imiquimod. It was a cream that she put on the surface of the tumor while giving paclitaxel, which in itself has immune stimulating properties. It was a phase 2 study, 15 patients, and what she found was very interesting. She had a 36% complete response rate, and these were confirmed by pathology. Here's this patient at the end of the study. You can see just a little scarring here. And when you biopsied the areas which were tattooed, you can see that the patient just has a nest of lymphocytes here, but no evidence of these tumor cells. The partial response rate was 36% with an overall response rate of 72%. Now, I can tell you, once we stopped treating with imiquimod, the patients had about 4 or 5 months before the disease came back. And we also looked at biomarkers and found that those patients who didn't have a complete response were more likely to have upregulated PD-1 on the surface of their T cells. And so we're very interested in following this up in this very unique patient population, giving this type of treatment along with an anti-PD-1 monoclonal antibody. So just to end up, that's T cells needed. When T cells are present, I do have to tell you, not only are we looking at more immune checkpoints such as TIM3 and LAG3, TIGIT, these are all in clinical trials as monoclonal antibodies, but we're also looking at using monoclonal antibodies to activate the T cells, CD28, 41BB, CD27. I can tell you that these have had some issues in terms of causing some very significant toxicity. So we're really learning the best way to give these drugs and sequence them in to a combination chemotherapy strategy. So I'd like to end with my final slide, just saying that there's much more to come. And if you looked at clinicaltrials.gov, immuno-oncology agents have had the fastest growth in clinical trials of any other agents, 233% more trials in 3 years. Whether they're a T cell targeted immunomodulator, some of these ones that modulate the microenvironment, cell therapy, cancer vaccines, almost 800 clinical trials of cancer vaccines in 2020, oncolytic viruses, and other types of T cell targeted antibodies. So I think we're just at the beginning of using the immune system to treat cancer. I know many people are hoping that we will have only immune therapy for cancer going into the future. I think we're a little ways from that, but we have many other exciting agents that are working their way into the clinic. And with that, can I answer any questions for you? Please feel free to use the chat, but happy to answer any questions.
We have a quiet group.
That's okay. We'll sit here for 5 more minutes. I don't mind.
Don't be shy. Any questions for Dr. Disis?
How do you guys like this seminar series? Is there anything you'd like to see more of? Karen, what do you think?
I think we have a question. Do you want to address the question?
In oncolytic virus therapy, how do you prevent the virus from spreading to non-tumor cells? That's a very good question. Many of these viruses are relatively short-lived. One of the issues with oncolytic virus therapy is, let's say you use it in hepatocellular carcinoma, which is kind of caused by a virus itself. Will it cause additional toxicities? So the oncolytic virus therapy, kind of like the CAR T cell therapy, is a bit short-lived. So you get your big bang for your buck, and the virus kills the tumor cells, and they're not fully replication sufficient, so they have a relatively short half-life. Why would the immune cells respond to tumor antigens after oncolytic virus therapy, but not before? That's a great question, Jessica. It's because the virus itself causes a danger signal. And when you look at the immune microenvironment in the tumor itself, the antigen presenting cells and the innate immune cells that are there, that are needed to stimulate the T cells, are not very functional. But when you put the virus in, the virus gives a danger signal to the immune system. and it changes the phenotype of those innate immune cells to a phenotype that presents antigen in a very efficient way, because your body is like, if I don't respond to this virus, I'm going to end up dying. And so basically now, even the T cells, these antigen presenting cells, have upregulated those activating, not the inhibitors, but the activators like CD28 and CD27. And the T cells are really getting a solid punch. So they develop immune responses. Right? It's one of the things, the T cells end up kind of killing the virus. But as the tumor antigens are released into this milieu, they're being more efficiently primed. Thank you, Ed, for those kind words. And please tell us if you have topics that you'd like to hear more about. We'd like to expand this series in the future, so let us know. So, is oncolytic virus therapy helpful in killing TMB low tumor cells? Not yet, Jessica. Those TMB low tumor cells, or those cold tumor cells, are a little bit of a conundrum. So they're still, oncolytic virus therapy is still more effective in the inherently more immunogenic tumor cells. Would stage 1 breast cancer be likely to benefit for which treatment? Right now, immune therapy is not being used for most breast cancers, but it is being used in a subtype of breast cancer called triple negative breast cancer. And many patients who have even early stage triple negative breast cancer are receiving immune checkpoint inhibitor therapy. So we're hoping we can find ways to expand breast cancer for immune therapy. It's such a common tumor that we'll be able to figure out how to make immune therapy work best for everyone. But so far, it's not a big winner in the immunotherapy lottery.
Any other final questions before we wrap up? All right. Well, thank you all for joining us today. Thank you, Dr. Disis, for your talk. I hope you feel better. Please do join us next week where we shift our focus more specifically onto cancer vaccines. Dr. Denise Cecil, who's a principal scientist here at the Cancer Vaccine Institute, will talk about how we go from a vaccine target to developing and testing a vaccine. So we'll see you next week on 30th, 3 to 4 p.m. Pacific time right here. Take care.
Bye, everyone.
Thank you.